Talk on evolution, which is called Evolution and Design. Just to say, I'm sure we all enjoyed Rabbi Dr. Freeman's talk last week. There's been a slight delay in uploading them to the internet, but as you can see, JB's busy going to be videoing this one, and they will both be going on the internet, so you can do Chazara, which we all know is very important to do review before the next one. Then there will be a 2-week gap until the next class. There's no class next Tuesday evening, so the next one is in 2 weeks' time on the 25th of February, by which time I'm sure they'll both be on the internet. You'll have been able to watch them both on the Shul website, and I think you'll start with a test in week 3. Okay. Of course. Thank you.
Thank you very much, Robert Lera, and thank you for coming back again. And it's a great pleasure and honor once again to be back here, particularly because I thrive on the questions that you ask and the debate that we can have towards the end. And I'm sure this will raise a number of questions. For those of you who were not here last week, I have a sort of a mantra which I try to drill into people, which is a concept that I learned from my own Rosh Hashiva when he was responding to a question on science and Torah. He said, if you want to discuss issues in science and Torah, you must ensure that you're learning both the science correctly and the Torah correctly, which was a bit of a light bulb moment, because I realized that for many people, they don't necessarily know the Torah correctly, they don't necessarily know the science correctly, or both in some scenarios. And while I have, I would say I'm certainly conversant in science, and I'm a rabbi, so it's my job to be an expert in Torah. I feel that I can help people to understand both sides. And once we can understand both sides, then we can start to ask the right questions from the right positions. Because otherwise, how can we hope to get any clarity whatsoever? So without further ado, once again, I'm going to give you a science lesson. And this science lesson is going to be this time on evolution. We're going to watch a short video as well to help us understand the origins of evolution. And most importantly of all, understand the progress that's been made within the idea, the concepts of evolution. And as we'll see, that evolution is not one idea, it's not one theory or one set of ideas, it's a complex set of ideas, some of which are provable scientific fact that have been repeated many, many times in laboratories, others which consist more of conjecture perhaps, but as we'll see, evolution is not to be sniffed at. It's not something that a person with a religious identity and with religious faith can simply sweep under the carpet, as some would like to do. So let's have a look at what we're going to do. We're going to look at the history of evolution, the mechanisms of evolution, the idea of common ancestry and descent, and the idea of neo-Darwinism, what neo-Darwinism actually is. So the history of evolution is as follows. You probably recognize at least one person on this slide, Charles Darwin in the middle, who was one of the most important individuals, personalities when it comes to evolution. Alfred Wallace was a colleague of his, someone who also worked on the science of evolution. You probably don't know who Charles Lyell is. Charles Lyell is one of my heroes because he started life as a solicitor and then he became a scientist. And he became, in fact, a geologist. He was fascinated with geology. And Charles Lyell, in fact, sowed some of the seeds that helped Darwin come to the conclusions that he came to. Now, Charles Darwin spent 5 years on a voyage on the ship called the Beagle, and he went all around the world. He went to South America in 1831, and he was reading Lyell's new book called The Principles of Geology. And there were 2 very important points which were moments for Darwin which helped him on his way to developing his theory of evolution. First of all, the idea of gradualism. Geological features can be explained by the slow, gradual action of processes we observe every day. The erosion of wind and water, the depositing of sediments in rivers, and so on. Secondly, and this was going to start riling the religious movements, the Earth is very old. And therefore, there has been a long time for these gradual processes to take place. Now, the route that the Beagle took went, as I said, all over the world, but it was particularly in the South American areas of the Galapagos Islands over here that Darwin made his big discovery. And his big discovery was through observing certain species of animals that he found on the Galapagos Islands. I'm going to play you a short video, and that video is going to help explain how Darwin came to his light bulb moment. Oh, except there's no sound. This is interesting. That's because...
Understanding of how Darwin came to his ideas in the Galapagos has been the myth of the finches. The idea that Darwin went to the Galapagos, he saw all these finches, he saw all the different beak sizes, and he had a eureka moment where everything about evolution came to him. It really didn't happen like that. He did see the finches, but he didn't really know what to make of them. It was another bird that he saw in the Galapagos that really started him thinking, and that bird was the mockingbird. Collected by Darwin on the voyage of the Beagle. These 6 here are the birds from South America, so Chile, Argentina, and you can see they're mostly sort of dull, brown, medium birds. Then over here we have 4 birds from the Galapagos, 1 from each of the islands that Darwin visited. When he came to the first of those islands, Chatham Island, he came across this bird, which he immediately recognised as being very similar to the birds he'd already seen in South America. But it was only really when he came to the second island, Floriana, that the real surprise was waiting. He saw this bird, and it struck him immediately that this is a very different bird. It's bigger, it has this dark chest, the bill is quite long, and what he realised is the differences he was seeing in the Galapagos between the birds were greater between birds from these islands very close to each other than really he'd seen over the whole of South America, and that's really made him start thinking.
Darwin's notes, the notes that he made about all his Galapagos specimens as he was sailing away from Galapagos. He writes about the mockingbirds. I have specimens from 4 of the larger islands. In each island, each kind is exclusively found. The habits of all are indistinguishable. When I see these islands in sight of each other and possessed of but a scanty stock of animals, tenanted by these birds, but slightly differing in structure and filling the same place in nature, I must suspect they are only varieties. The only fact of a similar kind of which I am aware is the constant asserted difference between the wolf-like fox of East and West Falkland Islands. If there is the slightest foundation for these remarks, the zoology of archipelagos will be well worth examining, for such facts would undermine the stability of species. And that is the point at which Darwin first suggests the possibility that species might change, the key insight for what was to become his theory of evolution by natural selection.
Okay. So what we find is this quote is probably the most central quote that we've learned from that. If there is the slightest foundation for these remarks, the zoology of archipelagos will be well worth examining, for such facts would undermine the stability of species. What Darwin realized was because of the variation on different islands between the same species, that it must be that because a particular species is confined to that island, that species on one island has changed in some way over time, gradually, over a long period of time, compared to the same species on another island. So within one species, we can detect differences that may be as a result of the environment that that particular animal is living in. So this is one of the diagrams of the finches. Here are a couple that you saw before. This was the finch the mockingbirds. This was the mockingbird that he saw on Floriana, and you can see the drawing is very different to one of the other mockingbirds that he saw. The famous picture that everyone will drag up is the image of the finches. As you heard in the video, it wasn't quite the way that it worked, but you can also see by looking at the differences in the finches that would spark someone to think that perhaps those changes happened as a result of the environment that these animals were living in. So together with other scientists at the time, Darwin began to work on why this would happen, and he showed that sexual reproduction provides the mechanism by which creatures can change within a species and form all sorts of different features. And in fact, he took the next stage and the conjecture, I suppose, which was that would therefore explain how all species exist, how all animals have come to exist, and why we have a variety of different animals. That from a simple single-cell life form, all complex organisms can come, such as fish, giraffes, and ultimately human beings as well. So he wrote The Origin of Species, and the 24th of November 1859, of course, this causes an enormous outcry, particularly among the religious community, but fundamentally he'd done a lot of research and he'd been working on it very hard, and therefore this idea was out of the bag, and it wasn't just Darwin that was working on it, many other scientists were as well. So what is the important feature of evolution? What's the important idea that underpins evolution? That is natural selection. And for that you need four principles. You need the idea of variation, that not all living plants and animals in a species are the same. There will be natural variation within a species. As we see from looking around the room, we see a natural variation between us. Competition. Okay. There's not enough food or space for all creatures and plants to exist. Okay. So there's competition within a species for food, for partners to produce children. There's also the idea of survival. Not all individuals within a species are going to survive, and therefore if you have features which will help you to survive, that's going to be a good thing. Reproduction, those creatures or plants which survive are more likely to have offspring. Naturally, if you survive for a longer period of time, it's more likely that you'll reproduce, and therefore the next generation will inherit the features that you have developed if you've developed positive features, and that hopefully will propagate on through the generations. So this was the concept, the mechanism which was driving this variation between different animals within the same species. So how does that work? We're going to do a little sort of demonstration of natural selection, and we're going to start with this green blob, which is sort of representing a creature of some sort. And this creature lives in an environment, a very sort of dark environment, where it could be hunted by a predator. And the darker the pigmentation in this green creature, okay, the more likely it is that it will be able to camouflage itself against the predators. Okay, so here it is. And this is going to give birth to 3 children, and due to variation, okay, this one is obviously going to have a slightly lighter pigmentation, and this one a slightly darker pigmentation. So given that this one is at a serious disadvantage because its lighter pigmentation means it's exposed to predators, we're going to imagine that this one is going to get eaten by some predator of some sort. Okay, so he goes away. So we're left with these two, and these two are now going to reproduce, and they also will have a certain amount of variation, and the same thing will happen. The predator will come along and will only be able to find the green blob creature with a lighter pigmentation. So these two now are going to disappear. Okay, and so on. So for the next generation, we've got these two. And you can see as we go through each generation, the fact that a darker pigmentation means a more likelihood of survival. You'll find that over time, that darker pigmentation will go through the generations and come out, and this creature, in fact, its pigmentation will darken over the generations. So that's the mechanism behind it. But how does it actually work on a molecular scale? So at the same time as Darwin, although it wasn't particularly well understood at the time, Gregor Mendel was doing experiments on heredity. And you've probably heard of Gregor Mendel. He did a lot of experiments on a variety of different things. He liked gardening. Pea plants, in fact, are the most famous thing he was known for. One of the words that we need to learn is the word allele. An allele is one or two or more forms of a particular gene or a group of genes. Sometimes different alleles can result in different observable traits, such as different pigmentation. So for example, there'll be an allele that will describe eye color or hair color, or whether a person's earlobes are attached or detached from the bottom. right? So there are all sorts of different alleles that geneticists will recognize, and Gregor Mendel did the first work to recognize that. Okay? He was looking at his pea plants, and he found that when he bred red-flowered plants with white-flowered plants, all of the offspring initially produced red flowers, each one of them, every single one, which was very strange. But then when he took that second generation, which had all produced red plants, and bred them again, he found that most of the offspring had red flowers, but some of them had white flowers again. So what on earth was going on? And he realized that, you probably remember this from school, right? These are the Punnett squares, right? He realized that the red gene, to have red pigmentation, right, was stronger, was dominant against the white one. So that means that within the first generation, each one is going to have red, but each one of these red plants is going to be a carrier of a white pigmentation gene. And so that when you breed these plants in the second generation, they will ultimately produce 75% of red flowers and 25% of white flowers. Okay? Now, it doesn't quite work that simply, because there are types of genes that in fact only have a partial dominance, and therefore you can end up with this sort of situation, and that's why it is possible, in fact, contrary to popular belief, to have two parents with light-coloured eyes and still have children with brown eyes, because it's actually, although the brown is always dominant, it can actually skip a generation in some ways. But anyway, we're not going to go into the details of it. The point is that he was the first person to realise that there was something going on, there was something being passed on through sexual reproduction that was allowing these traits to work. Now, neo-Darwinism is the bringing together of these two spheres. Darwin's idea that the variation was due to natural selection, and Mendel's seminal work on genetics. And of course now genetics has come a long way since the time of Gregor Mendel, and Professor Richard Dawkins is of course the most famous and perhaps the most vociferous proponent of neo-Darwinism, and his books and his work is very important. We should understand the concepts behind it very much. We should get to grips with it, so we're going to do that. First of all, there was another important stage that we haven't spoken about, and that was the discovery of the structure of DNA. Because we all know, we've all heard of DNA, but many of us will not realize how important the discovery of DNA, and especially the structure of DNA, was in understanding the mechanism of evolution. So Watson and Crick were credited with the discovery of DNA, although in fact it was others who did most of the important work. But Watson and Crick are the two famous discoverers of the DNA structure. And the structure of DNA is critical in understanding how evolution works. So let's give you a little crash course in DNA and genetics. Here is a cell, any cell in your body. And within that cell there are all sorts of different structures, most of which are not drawn on this diagram. But the important part of the cell is the center of the cell is called the nucleus. And within the nucleus we have a number of chromosomes. Each chromosome contains, wrapped up inside it, DNA. Okay, this is the DNA over here, and you can see how it's all wrapped up very, very, very small. And this is probably very familiar, this image of the DNA double helix over here. Now, genetic information is encoded in this DNA through three nucleotides: guanine, adenine, thymine, cytosine. The names are not terribly important, but they're recorded using the numbers, the letters rather, of the first letter of the name, G, A, T, and C. And these make up pairs, base pairs within the DNA. And it's the order of those base pairs which will define what that DNA molecule does and what it creates, what it makes, and therefore define every single feature in your body, whether it's your height, your eye color, your hair color, everything about you. In fact, whether you're male or female, everything is going to be encoded in that DNA. And that means that potentially one could take any cell in your body and reproduce you as a human being from that one cell, because all of the information is contained in each and every cell. So here are the 23 chromosomes that human beings have. Other animals have a different number, but here they are. And in fact, the 23rd are known as the sex chromosomes. If you have an X and a Y, you are a man, and if you have an X and an X, you are a woman. And the determining factor as to whether a child is born male or female is in fact the male seed rather than the female, and therefore that's why Henry VIII was unfortunately wrong. It wasn't the fault of his wives that weren't giving him sons, it was actually him, although apparently he fathered many other sons who were from extramarital relationships. But nevertheless, right, if it's a boy or if it's a girl, that's our fault, not the wife's fault. Right? So here we go. Here is a scanning electron micrograph of the same chromosomes over there. You can see the sort of exclamation, and if you were to unpeel these, you would find all the DNA inside. Okay, so how does it work? Well, we mentioned at the beginning that Darwin realized it was sexual reproduction that allows for these variations to take place. So what actually happens during reproduction? So as a cell divides, there are different ways that a cell can divide, first of all. The important way that a cell can divide in terms of the production of sperm cells or egg cells is a process called meiosis. Okay, so a sperm cell or an egg cell inside a man or a woman respectively, these are called germ cells, or in the biological parlance, a gamete. Okay, gametes. So what happens? As the cell divides, copies of the genetic information are made, and each cell then divides twice to form 4 gametes. So you end up with a process by which each chromosome is split into, is divided into 2, so you end up with a copy of that chromosome, and then because it has 2 parts to it, that's then stretched apart, and you end up with 4 parts, and you end up with 4 cells. Okay? Well, let's show you a little demonstration. Here we go. Here are 2 chromatids, so that's 1 chromosome together, and the reason that I've drawn them blue and red is because each one of us, as you'll see in a moment, each chromosome that we have, we get 1/2 from our mother and 1/2 from our father. Okay? So each one of us has 23 pairs of chromosomes, right? In every one of our cells. Each pair contains the chromosomes from both of our parents. Right, so let's go on. What happens? So the first stage is that set of DNA, that set of all those chromosomes are going to be copied. Okay, so you get a copy. That's the first stage of cell division. Then as the cell divides further, the homologous chromatids, which means the chromatids that contain the same type of information, come together, and information in sections of these chromatids starts to swap over. Okay. Starts to swap over. Very interesting. There's a sort of mixing of the DNA. Then what happens is that these 4 chromatids are separated, and these then go into making up either 4 sperm cells or 4 eggs. Okay? Now, the reality is, you should know that this process is going on constantly within men. We are always producing more and more sperm. However, in women, all of the eggs that a woman will ever have are in fact contained within her from the very beginning of her life. Okay, I remember hearing from Rabbi Tatz that he once, when he was starting to be a doctor, opened up the ovary from a young child, and all of the eggs were there. Okay? They're all contained from the beginning. So obviously what happens during sexual reproduction, excuse the diagram, is that the sperm enters into the egg, and these 2 chromatids then come together. And of course this is happening 23 times for all of the chromosomes that you have. Okay? You then have, therefore, a set from a chromatid from your mother and a chromatid from your father for each one. But you can see as well that the information has all been mixed up. Okay? This then divides and keeps dividing, and of course eventually it makes a little baby. Okay? So that's how it all works. Now, that's all very fascinating. Where does all this fit in when it comes to natural selection and everything else? So here I put a made-up code of DNA. Okay? So you remember we talked about those 4 bases, the C, the G, the A, and the T. So this is a code, and this code will code for a particular set of proteins or whatever it happens to be doing. It might code for your eye color or your hair color, whatever it is. One of the things that Watson and Crick discovered, which was possibly even more important than the actual structure of DNA, was that they worked out how this code is actually translated into proteins that each DNA section will actually produce. And what they discovered was the following, that this code is read in triplets. Okay? Here are the bases. Each letter is a base. And here are the triplets that we call codons, because they code for a particular amino acid. Okay? These amino acids can be calculated and be worked out in the laboratory what each codon actually codes for. And together, these amino acids form to make proteins. And it's those proteins which then build a human being. Okay? That's how it all works on a very basic scale. That's all well and good, and here you have, in fact, a graph, a diagram, I suppose, of all of the different combinations of all of the different bases and which amino acids they produce. Okay? So we can work out from the DNA code what's actually being produced by that code of DNA. So DNA produces amino acids which produce proteins. Now, the way that a protein is produced is very important, because that code will affect exquisitely what that protein actually does. This is a picture of a protein, a diagram of a protein, and you can see it's a very complicated molecule. So this is the actual code for this particular protein over here. You can see all the amino acids that go in to make that. And you can imagine that if one of these amino acids was different, that whole protein would look different. It would be a completely different protein. So what happens? What's the theory? The theory is as follows. And this is actually, as when I said some of this is scientific fact, this is provable beyond any reasonable doubt whatsoever. What can often happen is that during the division of cells, and this can happen in all cells, not just through meiosis, but also through the other way that cells divide, which is called mitosis, okay, which is when regular cells divide. What can often happen is that we're supposed to get this, okay? This is cell division going on over here. We're supposed to get an exact copy of that information. But what can happen for all sorts of different reasons is that as that cell division occurs, we can end up, for example, with one of these bases being knocked out. Or these bases can actually change. They can change from G to C or whatever it happens to be. But let's imagine for the moment this base over here, this little G, has been knocked out and disappears completely from the code. Okay, and that can happen because of a variety of different reasons. There can be mutations, natural mutations. There can also be, it can also be affected by radiation. Radiation can affect that, 100%. That's why radiation can cause cancers. Some cancers can be caused by that. That's also why skin cancer in particular is caused by ultraviolet light, because ultraviolet light can affect the division of cells. So therefore, if you have cells on the surface of your body which are dividing quite rapidly, ultraviolet light can knock out, can mutate various cells. And if they mutate in a certain way, they can become cancerous. That's the mechanism behind it. But what then happens? So you've now got a modified code. And remember that we said that Watson and Crick discovered that this code was read in triplicate. Okay, sorry, not in triplicate, it's read in codons which are made up of 3 bases. So that means that this one's fine, because that's the same as it was before. But now, as you go through, each one is going to be shifted to the left. So instead of reading GCA, we're now reading CAG, TGG, GGA. Okay? And as a result, there you go, that is then going to produce a different set of amino acids. And therefore, because it's producing a different set of amino acids, a different protein. Now, it could well be that that this cell will in fact mutate in such a way that it can't survive, it won't work anymore, and it will simply apoptose, it will simply destroy itself, which happens quite regularly. But every now and again, bad things will happen. Often as a result, we'll get perhaps cancerous cells, which will start dividing uncontrollably. That's what essentially cancer cells are. Okay. So this is what's going on over here. Now, the theory is that mutations in cells, natural mutations, can also produce positive effects. And it's that positive mutation that results in, for example, the greener pigmentation, or the darker greener pigmentation that we saw in our little model that we made before. And the variation that we see in species is not just because of the mixing of DNA, but because of these natural mutations that occur all the time. And as a result of that, this is what's driving the whole of evolution, and the reason that different creatures exist today. That we see a whole plethora of different species in the world that could have all come from a single cell is because of random mutations that can take place that eventually will manifest themselves as positive outcomes for a particular species, which will be maintained because of natural selection. So we see mutations occurring all the time in nature. If we say that each gene is the instruction for making one protein, that mistake is going to be copied over and over again, and it'll end up making a different protein. So for example, albinism, okay, which we see in all sorts of different animals. Here we have an albino tiger, okay, that's very fascinating. Right? It's a mutation, it's a genetic mutation, that's what's happened, okay? However, and this is going to be the sticking point, where a scientist, if he's worth his salt, will admit that most mutations, in fact, are destructive, okay? I've given you an example here of bacteria, okay? Now, bacteria don't reproduce sexually, but the point is that we can see that mutations here can actually cause problems, right? So in this particular scenario, because bacteria divide so rapidly, what we see is that they can mutate randomly to the point where they become resistant to certain antibiotics. So that's why there are bacteria today that are resistant to antibiotics that we used in the past. And that's also why the overuse of antibiotics in fact causes us problems, because eventually bacteria can become resistant. That's why you end up with things like MRSA, methicillin-resistant Staphylococcus aureus, which unfortunately can be picked up in a hospital and can be very dangerous to a patient perhaps who's very unwell. In fact, we probably carry the bacteria on us all the time, it's just we're not affected by it. Sickle cell anemia is another type of disease that's a result of a genetic mutation. And interestingly, although a person who is in fact suffering from sickle cell anemia would be very sick, would be very unwell, a carrier of sickle cell anemia in fact is protected from certain things like malaria. So we can see that there are certain positive advantages to random mutations, but most of our experience in the scientific world is that mutations are destructive. Nevertheless, that is still the basis for neo-Darwinism and evolution, and that shouldn't be sniffed at, because there could well be good reasons why we generally see negative and destructive mutations, and why there still could be the mechanism behind evolution. What about common ancestry? The descent of man, as Darwin put it. He publishes The Descent of Man, and of course it shocks Darwin's opponents, because he's now saying that, we all descended from monkeys, what's this all about? It's widely accepted in the scientific community, and I think we can all appreciate that within a species there was a sort of descent from an original paradigm species. Okay, so all breeds of dog we know came from a single breed, from in fact the wolf, right? In the scientific world, the evidence for common ancestry is very strong, and I'll show you some of that evidence right now. So first of all, we have the idea of homologous similarities between species. We have a fossil record which will help to point in the right direction, although it's not clear, it's not 100% clear. And we also have what's called vestigial structures in embryology, which show that certain creatures have structures that appear to be left over from a sort of like previous incarnation along its evolutionary path. So what are homologous structures? Well, we see, it's fairly obvious. If you look at different animals, they are essentially very different and have very different structures and sizes and everything else. There are, in fact, enormous similarities between the general design of a limb. So a whale, a cat, a horse, human, a bat, a bird, we all have very similar structures, even though some of them will be emphasized in different ways. So a bat has a particularly long set of phalanges. In ours, our fingers are fairly short and stubby and they don't have skin between them, we can't fly with them, but you can see that this implies that there was a common ancestor, a common design which has varied in different directions. Similarly, going back in time, we can also see, you've got a pterosaur over here, a bat, and a bird. You can see a similar structure and a similar design to each one. Also, there is an overall trend in the fossil record. Now, it's very important to note that the fossil record can't be complete, we can't see everything, but that overall trend indicates that as life started on Earth, it started with very simple-celled creatures that then progressed onto to more complex things. Okay, so here's a nice little diagram. It's probably not terribly clear, but you can see that we start off with much more simple creatures, trilobites and all sorts of tiny insects and things, and we eventually come up to the sort of dinosaurs that you're probably familiar with until we end up with mankind right over here. Okay. Vestigial structures, such as these pelvic bones in the baleen whale, appear to show evidence for a sort of previous incarnation along the evolutionary path. The baleen whale, in fact, started as a land creature, and this pelvic bone over here appears to do absolutely nothing whatsoever. Okay, there are things that scientists once thought were vestigial structures and evidence of evolution. This certainly appears to be that, and the baleen whale carries these sort of bones that appear to be nothing, which indicate that it once had limbs coming out of here that have now disappeared. That's just one example of many. Right, so you're probably all familiar with this diagram. I'm going to give you a little light-hearted break just to sort of clear the fog a little bit. You probably haven't seen that diagram yet. Okay. So this is one of my favourites, actually. There's another one, and this is one of my brother-in-law's favourite. Now, the truth is, however, that this is where the bigger problem is going to come. Okay, we'll get to working out the Torah approach to all of this in a moment, but the key that I want you to take home from this slide is this number over here, that Homo sapiens sapiens, which is us, our species of animal, so to speak, has existed for around 200,000 years. Okay? Burn that into your brain for the time, because that is going to be very important, critical in fact, to understanding and to working out how we can relate all of this to Torah. So, to summarize what we've just said, we've got the fact that the concept of evolution involves a number of complex steps. There is strong evidence for natural selection within a species, and we know the mechanism which allows natural selection to take place. Darwin hypothesized himself that all life, including man, has evolved from single-cell life forms. Cell division and mutation of DNA are both observable facts. We know that that happens. Evolutionary biologists claim to have found examples of speciation, which means one animal will sort of eventually turning into different species later on. Okay? Now, some of the criticisms of evolution are very important, and we should give them a little bit of time. Studies on the effects of genetic mutations indicate that they are destructive events, which we've spoken about before. At best, the information stays for some time, but often harms or kills the organism or cell. We don't have any examples, many examples, perhaps even any examples, of where random mutations actually add useful information, which would yield new structures or organisms. Also, the mutation rate is in fact very low. Okay? So, of these, beneficial mutations make up a small fraction, and the observed beneficial mutations are not the information gaining type needed for evolution. That's another criticism. The smaller the change, of course, the smaller the selective advantage. But the smaller the selective advantage, the more likely that random effects will eliminate the organism. So, for example, even if you get a positive mutation, you've only got approximately a 1 in 500 chance of surviving, and that is then passed on to the next generation. Even if a beneficial mutation survives, for it to become fixed in a population, the organisms not carrying it must be eliminated as well. So if you want to read up on this a little bit further, and some of the main criticisms of evolution from a mathematical perspective and from the rate of mutations, I would suggest reading Dr. Lee Spetner's book, Not by Chance, and it deals with a number of these issues in that book. I would also suggest that that doesn't mean that we can, again, sweep evolution under the carpet and say it's a load of nonsense, because, as we've seen, there are a number of complex ideas which relate to one another, some of which are very much provable and observable. The question is, what do we do with all of that? Because the problem that we've got is that in the olden days, we used to sort of say, well, science can't explain everything, so that must mean that God exists, and that's where God is, right? The problem is that science came along and started to explain all of those things. So that means that God can't exist anymore, right? Because we've explained it all. So often you'll hear secularists and scientists such as Professor Dawkins say, well, we don't need God to explain all of these things. In the olden days, before modern science, man had to come up with answers to the questions as to how life had come to Earth. Why do we have different animals and plants? So he made up all of these interesting stories, and every culture has its own set of stories as to where all of these animals and plants came from. The Torah is no different. The Torah is also just a set of myths, chas v'sholom, that were made up in order to explain the existence of all sorts of animals. And if you read the Torah like a 5-year-old, as we spoke about last week, then that's the conclusion that you'd come to. well, you know, God created the heavens and the earth, and there was a garden, and there were trees, and there were fruit, and there was a man, and there was a woman. Come on. So, from a scientific perspective, evolution is there to replace God in the sense that it explains all of the life that we have on earth. From our perspective, though, God was never invented in order to explain the existence of life on earth. God was a fact and something that we have faith in from a different perspective, which we'll explore in one of the coming talks. The fourth talk will explore the nature of faith and why it's different to science. But for the time being, we've got a number of questions. First of all,